Hi everyone, I'm Rhett Smith for ProtonGuru.com. I'm going to talk today about some MCAT preparation. And in today's video, I'm going to solve the Chemical and Physical Foundations of Biological Systems section of our free full-length MCAT exam. So once you've logged on to the full-length MCAT practice course, you'll see a variety of different choices. You can see, first of all, the instructions for preparing for and taking a practice MCAT exam. Essentially, this advises you to do this in sort of a situation that will mimic the real exam. And today we're going to be looking at specifically the Chemical and Physical Foundations of Biological Systems section. This section of the real MCAT has 59 questions, and you'll have 95 minutes to complete them. So you can set a timer to check your pacing, or you can go through and do the practice mode to just read the explanations as you go. Either way you want to do this practice, of course, we'll be able to do that. So there are two things you're going to have. You're going to have two separate windows. One will be the passages, and one will be the actual quiz questions. On the real MCAT, you'll also have the ability to see side by side the passage with the questions like this. And also in the real MCAT, you'll have a periodic table resource file. I'm just going to open that and leave it in the background. You'll be able to click it open and close on the real MCAT as well. So I open up the quiz and it will show the first question, but I wanted to point out that you have this side dashboard that you can pull up at any time. The way you toggle that off so you can just focus on the exam is to do this. So we can open and close that. Alright, so now I've got the passage file pulled up. I kind of like to see the whole passage if possible. Some will be a little too long for us to do that with a reasonable size font, but I like to see the whole passage if possible and the question at the same time. The question will always tell you which passage you should be looking at as well. And this passage is here. It also tells you how many questions you have to get through for that passage. So 5 questions on this particular passage. And you can download the file from the site and read it on your own. But I want to go through today and just talk through solving the questions. And of course you can go over here and highlight different things if you notice them as you're reading, so that you can toggle over to this screen, highlight some other things, and think about things while you're answering the questions. But here's our first question. When mutation occurs to hemoglobin A that causes a change in the surface charge of the protein but not of the protein's molecular weight or size, which of the following techniques would be most likely to achieve good separation of the mutated from the native hemoglobin A? And we don't necessarily have to read the passage about the mutated hemoglobins to get this question right, because it really is a question checking our knowledge of the different ways to separate proteins. Size exclusion chromatography separates by size. Centrifugation does as well. The sodium dodecyl sulfate PAGE, STS PAGE technique, will sort of add these STS molecules, negatively charged, to the surface of all the proteins in the sample so that you sort of even out all the charges. So that is not what we want in this case. It's really only the isoelectric focusing technique that separates proteins on the basis of their charge, not on the basis of their size. So if you're in a practice mode on this type of course in the MCAT Ladder, you'll be able to look at the individual explanations as you go, or you can just skip the answer and go on to the next question. So question 2 is asking us if atmospheric pressure is 760 torr and the air is composed of 80% nitrogen, 15% oxygen, and 5% other gases, and assuming the presence of nitrogen and other gases does not influence Kd, which is true of the hemoglobin A plus O2 reaction that's shown in the passage. So that's referring to this reaction right here. And if you read this passage, it says that the Kd for this binding of the first O2 is 180 torr, meaning that to reach equilibrium you need to have at least a concentration of O2 that reaches that pressure. Here the pressure total is 760 torr in this question, but we're being asked for the concentration of O2, or we need the concentration of O2 to see what the equilibrium is. So we need to find the partial pressure of O2. 15% of the total gases is oxygen. That means 15% of the pressure will be the pressure of oxygen. So when you do the MCAT, you have this whiteboard with a marker, so you have to be able to write, with a relatively heavy pointed pen. So I'm going to write 760 torr, and only 15% of that is what we can count for the O2. So 15% is better written as 0.15. We have to do this math without a calculator. So 0.15, I think of as 0.1 and 0.05 added together, right? 0.1, 0.05, it gets 0.15. And that makes the math a little easier because a tenth of 760 is 76, and 0.05 is half of that. Half of 76 is going to be 38. So the 0.1 plus the 0.05 of the 760 lets me quickly calculate that 114 torr is the pressure of the O2. All right, 114 torr. We have to remember that as we go from our whiteboard back to our question. So the problem tells us that we need 180 torr of O2 to reach equilibrium. So we don't have enough O2 to push the reaction to favor the right side. So the left side is going to be favored for a native hemoglobin having those metrics. And the question has two choices, C and D, that both say the left is favored for the native hemoglobin. The other part of the question asks which side would be favored for the mutant hemoglobin. Now the information we have about how the mutation influences the binding of iron is given to us in the passage up here. Mutations can lead to changes in the surface charge as well as to changes in the binding strength of iron to oxygen. So the passage tells us that the binding strength can change, but it doesn't tell us whether the oxygen binds more strongly or less strongly to the iron. So we don't know whether the mutation will need more or less oxygen pressure. So we have to choose an answer that reflects that. The reaction is favored to the left only for the native hemoglobin, but definitely not for mutant hemoglobin. That's not something we know for certain. The reaction is favored to the left for native HbA, true, and possibly for some mutant hemoglobin A. That is true. And you can confirm that and get the right answer here in practice mode. Now we go on to question number 3. Here's a quote from the passage. The statement that hemoglobin A, quote, consists of 4 domains, 2 alpha chains and 2 beta chains, is most specifically a description of the protein's what? Here's a case where we don't really have to look at the passage to figure this out. We know that the quaternary structure is what is typified by the assembly of different domains together. These other levels of structure refer to, for example, the amino acid sequence and things of that nature. So the quaternary structure because of the counting of domains. Question 4 asks which of the following is a correct term for the heme unit in hemoglobin. Now you probably know that the heme unit is the unit that has the iron bound to it and allows the protein to bind oxygen because it's such a common protein. But it does mention in this passage what the role is as well. You probably also know that the heme is not made out of amino acids. It's made out of a unit called a porphyrin that binds the iron. When you have a group like that, you have what is called a prosthetic group. It's a group that is not made of amino acids, but it is still necessary for the enzyme or protein's function. Because hemoglobin has an iron bound to it, it is mentioned in the passage that you can use a radioactive isotope of iron, iron 55, to study hemoglobin. So in a study on hemoglobin A, a scientist measures the rate at which iron 55 decomposes to manganese 55. And the question is, by which process does this degradation occur? This falls under the nuclear physics or nuclear chemistry aspect of the MCAT outline. So we quickly toggle out to our periodic table, and if we're going from an iron 55 to a manganese 55, we note that the change is only in the atomic number. The mass is still 55. So we're going to have to lose a proton somehow. So our task is really to figure out which process does that. Well, in alpha decay, you lose an entire helium nucleus, which is a loss of mass. We know we don't lose mass, so that's not going to occur. Beta decay involves a neutron becoming a proton. That would cause the atomic number to go up by 1, but the process they're describing has the atomic number going down by 1. In gamma decay, the mass doesn't change, and the element doesn't change. It's an energy state that changes. Electron capture, however, involves an electron essentially combining with a proton to make a neutron, which means the mass wouldn't change because a proton and neutron are about the same mass, about 1 atomic mass unit, but you'd have 1 fewer protons. So instead of iron, you have 1 fewer in the atomic number. That's manganese 55. So electron capture is what we would expect in this case. So here we move on to passage 2. This will be for questions 6 through 10. And you'll see that this passage has some information. You'll often have a graph or a table or numbers in the passage on which you'll have to base deductions about experimental results. And here we also have some chemical structures. And again, I do like to look at the question before I necessarily comb through the whole passage. So let's look at this question. Which pesticide would be most likely to exist in a cationic form in water at pH of 6? All right, a pH of 6 is a little bit lower than neutral 7. Lower than 7 is acidic. So we have an acidic solution and we have these pesticides. Perhaps the most familiar instance of a slightly acidic or neutral pH leading to protonation of an organic compound is where we have amino acids. You probably know that the amino acid's amine nitrogen group is protonated even at neutral pH. So in looking at these structures and seeing amines on atrazine, hopefully we would sort of think about the amino acid and say, okay, we know that that's protonated. So definitely the atrazine would be the protonated one at an acidic pH. Now if you've taken a second to read the passage, you know this is about extracting these different pesticides from food materials. And this question is about the extraction process. Given that the extraction occurs at 372 bars of pressure, which best approximates the mass of carbon dioxide used for the optimized extraction described by the researchers? So when you have a passage like this, a good strategy when they're asking about a specific number that's in the passage is to graze through the passage and find just that number. So 372 bars of pressure. So here's where they're talking about it. And the sentence of interest is right here. And this is where they talk about it. They say that they use 29 milliliters of supercritical carbon dioxide having a density of 0.90 grams per milliliter. And they're asking us for a mass. So as soon as we see that they're giving us the volume and the density, it's really a question of using that density and volume to figure out the mass. 29 milliliters, 0.9 grams per milliliter. So we shoot over to our whiteboard, pick it up, and we say, well, the density is 0.9 grams per milliliter. We have 29 milliliters, and we have to get the answer. Right, the milliliters is going to cancel nicely, but without a calculator, 0.9 times 29 is not super easy math. So I would recommend doing 0.9 times 30. So 9 times 3 is 27. So going over here, it does ask us which best approximates the carbon dioxide. We estimate it would be 29. 26 is closest by far to the answer we got. Turns out that is correct. Here is another question concerning the extraction. Which of the following biological components would be most readily extracted if hexane was used as the extraction solvent as described in the passage? So instead of using carbon dioxide, they're going to use hexanes. And really we don't need the passage necessarily to answer this. We are saying if hexane is used to extract from a biological system, which compound would be most soluble in hexane? So one thing we need to know is hexane, being an alkane, is only carbons and hydrogens. There are no polar bonds. It's a really, really non-polar solvent. So it should be good at dissolving non-polar things, like dissolves like. So now it's really relying on our knowledge of the structure of these different biomolecules. I mentioned a little bit ago that at physiologic pH, where you'd be extracting these things, amino acids are zwitterions. You've protonated the nitrogen and you've deprotonated the carboxylic acid. Ionic compounds are really polar. They're not going to dissolve well in a non-polar solvent like hexane. How about carbohydrates? Well, we don't need to know a specific carbohydrate to know that carbohydrates have tons of OH groups on them, generally, a bunch of OH groups. That's really polar. Probably a really good hydrogen bonding solvent. Polar protic solvent would be good at dissolving carbohydrates. Definitely not hexane. Nucleic acids, we know, have this phosphate backbone that's actually carrying a negative charge. And again, ionic things are not soluble in really non-polar solvents. How about cholesterol? We talk about cholesterol when we study lipids in the organic chemistry and the biochemistry courses. And this is a relatively non-polar molecule that can embed itself in the non-polar region of lipid membranes. So that's definitely the best option for dissolution in a really non-polar solvent like hexane. Here's another question about the extraction, and this one mentions that effective extraction of pesticides relies on access of extraction solvent to spaces in a biological structure where the pesticides are present. If supercritical CO2 flows at a rate of V1 and a pressure of 372 bars through a circular tube of diameter 4 micrometers, what will its flow rate V2 be after being forced through a circular tube of diameter 2 micrometers? So it sounds pretty complicated, but they're giving us a lot of details we don't need. In the end, all that's happening is the fluid is going from a tube with this diameter, 4 micrometers, to a tube of half the diameter, 2 micrometers. So we need to engage our knowledge of fluids from physics, really a high-yield MCAT thing to study. So we grab our whiteboard again, and we think about the continuity equation, or the fact that the velocity with which a liquid flows through a tube times its area can't change if I constrict or expand the area of the tube through which it's going. So I've got to squeeze the same volume out, no matter how narrow that opening is. So conceptually, if I just imagine a hypodermic needle, if I push the plunger down on a hypodermic needle so that I squeeze out 1 centimeter, cubic centimeter, that will come out at a really high velocity because I have to increase the velocity to get that same volume through the same amount of time to flow through a tiny needle. So that kind of makes sense in helping us remember the equation, that it's a constant. Well, how does that relate to this particular problem right now? Well, we're trying to solve for V2 in terms of V1, so I'm just going to isolate V2 on one side. And it's just a matter of knowing how the area changes of the tube through which our fluid is flowing. So the area of a circular opening, or the area of a circle in general, is pi r squared. So we need to quickly figure out how the areas differ in this case. Area 1 is pi, radius is 2 squared, right? Diameter of 4 is a radius of 2. Area 2, pi, the radius is now 1, because the diameter is cut to half to be 2. 1 squared is 1, 2 squared is 4, pi, pi. So we need to plug these in. A1 is 4 pi. A2 is just pi. It's 1 pi. Pi will cancel, so V2 equals 4 times V1. This is one of our answer choices, choice D, and that is the correct answer. So now we look at the HPLC trace. In the HPLC trace, the most polar compounds are eluted first due to the experimental conditions selected by the researchers. Given this information, which compound most likely corresponds to the peak labeled 1? So look over here. Here's compound 1. All right, compound 1 is what is eluted first. They say the one that elutes first is most polar. And there's information about how they do the extraction and HPLC analysis in the very last sentence of the passage. The pesticides were extracted from acidic food juices and then separated for analysis without further modification using HPLC, trace B, that we see here. Now, under acidic conditions, we talked about the fact that atrazine will be protonated, but the others will not. And the ionic compound will certainly be the most polar. So peak 1 should correspond to atrazine. And that is the correct answer. Now we move on to passage 3, which will cover questions 11 through 15. The number of questions you may have noticed isn't the same for every single passage. So when you go to the MCAT, make sure you sort of budget your time according to how many questions you can get out of a given passage. So here's this passage, and you could pause and read it. Of course, it's easier to read if you download the file from the course site, which you can get this exam for free. And this question says, which is most likely to be involved in biofouling by mechanisms alluded to in the passage? So we have to sort of look at the passage for the mechanisms by which it says biofouling occurs. And the definition we need is right in the first sentence. If you don't already know what biofouling is, it says that nonspecific protein adsorption and cell adhesion is biofouling. So knowing that and looking at this, we're looking for a protein, of course, because that's what's going to do the adsorption according to the definition here. And we see DNA ligase, integrin, antigens, and ketose. Well, ketose is a carbohydrate, so we rule that out immediately. Antigens, integrins, and DNA ligase are all proteins. So what would be the best answer? Well, the best answer would be one that is capable of non-specific protein adsorption and cell adhesion. Enzymes and antigens are proteins that tend not to have very non-specific interactions with other biomolecules. Antigens and enzymes like DNA ligase have very specific binding events. So they're probably not great answers. Integrins are cell adhesion proteins that don't have this great specificity that DNA ligase and antigens have. So although antigens and DNA ligase could hypothetically be involved in biofouling, some non-specific interactions, you're always looking for the best answer on the MCAT. So integrins are the most likely to be involved in such a process. Next, we're asked which of the following forms of electromagnetic radiation could be useful in place of UV light to facilitate cross-linking by the mechanism described in the passage. So we're going to go over to the passage and look for where they're talking about cross-linking. So we see the word cross-links here, and it's also easy to pick out the words UV in a couple places because UV is capital U, capital V. So we can quickly kind of pick those things out. So if we read this little section, photocrosslinks, covalent bonds made due to electromagnetic radiation exposure to several substrates upon UV irradiation. When the phenylazide groups are exposed to UV irradiation, they're converted into reactive nitrines that can be inserted into CH bonds. All right, so somehow we're breaking CH bonds and doing a insertion into that bond. Not a reaction we learn about a lot in organic chemistry, but it's telling us this happens with UV light. So the question is, which other radiation could accomplish the same feat? This is really asking us to evaluate the energies of these types of radiation. UV radiation, we know from our study of UV spectroscopy, is capable of pulling an electron out of a bond. That makes sense because they're telling us that you're inserting into a bond, so you must break the bond to stick something into its place. So we're really looking for something with enough energy to do that. We kind of know that infrared radiation doesn't break bonds because in infrared spectroscopy, when you shine infrared light on something, it makes the bonds vibrate and bend but not break. Radio waves are even lower in energy on the electromagnetic radiation scale, so even weaker. And we use radio waves when we do nuclear magnetic resonance spectroscopy. Microwaves are also lower in energy than UV waves. So none of these have the energy necessary or appropriate energy to break chemical bonds. So a lot of this passage, if you've read it, is about adhesion, and biofouling is a type of adhesion. So this question says, for strong adhesion, a particular biological substrate requires both hydrogen bond donors and acceptors be present in the polymer backbone. Which polymer would form a strong adhesion to this particular substrate? So again, it's not really one we need the passage to answer. We have this ionic polymer like this, polyethylene glycol, polyglycerol, or none of these. Now, we aren't expected to memorize polyethylene glycol and polyglycerol necessarily. It's getting ready for the MCAT. But glancing up at the passage, they actually give us the structures of these materials. So we have the structures at our disposal, but we need to know the definition of a hydrogen bond donor and acceptor. The donor has to provide the hydrogen, and the hydrogen has to be quite polarized. So it's generally a hydrogen attached to a really electronegative atom, nitrogen or oxygen. So that would be what we call the hydrogen bond donor. The acceptor will be a really partial negatively charged, usually oxygen or nitrogen in the case of biological context, that you'll attract the hydrogen bond donors to. Even if you don't remember which is called the donor, which is the acceptor, you're looking for the OH or NH and then the O or the N. You need both in this case. And we see we have some acceptor groups, some polar oxygens in this case, but we don't have any hydrogens attached to a oxygen or nitrogen to do the hydrogen bonding. In ethylene glycol, the structure here, it's a polyether. So we have again a polar O, but not a polar H. The polyglycerol, on the other hand, has the OH group in its backbone. The OH contains both the H in polyglycerol and the O, as well as another O in its background that we could have as an acceptor. Next, we're asked how many non-bonding electrons are found on the nitrogen atom in a nitrine. Now, we probably haven't read about nitrines in our 2-semester organic chemistry class, but the passage defines what a nitrine is. It refers to reactive nitrines right here and defines them as neutral species with a phenyl group on a nitrogen. So we go over to our whiteboard and we put a phenyl group on a nitrogen. It has to be neutral, according to the definition in the passage. A neutral nitrogen atom, if we look at the periodic table, it's in the tall column 5, has 5 electrons. So that is the number of electrons we need for a nitrogen to be neutral. In this chemical bond, there are 2 electrons, and we kind of say that nitrogen owns one of them in counting for formal charge. So I need 4 additional ones around the nitrogen in the nitrine for it to be neutral, to have the 5 total. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Remembering that it only sort of owns half the electrons in a bond because it's sharing one with the phenyl group. So this question asked us how many non-bonding. So all 4 of those, 1, 2, 3, 4, that we had to draw in aren't doing bonding. So jump back to our question, put down our whiteboard, and we see that there are 4. Next, which of the following would be a Polyzwitterion at neutral pH according to the definition used in the passage? So you may not know what a Polyzwitterion is. We go to the passage and try to find that word, Polyzwitterion, and they say that Polyzwitterions are defined here as polymers with repeating units having 1 positively charged and 1 negatively charged functional group. It's a very specific definition, so it should be relatively easy to find something like that. And we have 2 choices, and then neither of these, both of these type answer selection. A polypeptide made of aspartic acid residues only. A polypeptide made of serine residues only. And this is really them sort of trying to trick us, because aspartic acid and serine are both amino acids. Let's go over to our whiteboard. So we're looking for a Polyzwitterion, and it's tempting to use an amino acid because the amino acid has a zwitterionic form like this. But remember, they're really asking us about a polypeptide. If you make a polypeptide, you have amino acids strung together where you've made amides, not ionic at all. So a polypeptide is not ionic unless there's some charge on the side chain. So they mention using aspartic acid. If you use aspartic acid or glutamic acid or lysine, something with an ionic side chain, you can get a charge down here. And I'm just drawing generically a chain. That's not the correct length. So that would be possible to have a charge down the backbone, but it doesn't have both a plus charge and a minus charge down the backbone in the primary structure. So neither of those potential choices is actually a Polyzwitterion according to the definition in the passage. We jump back up here, put down our whiteboard, and plug in that neither of those is the correct answer. So now on to passage 4. This is about gene delivery systems and different ways that can be accomplished. And our first question related to this passage is which of the following would be the best candidate for delivering RNA via the routes discussed in the passage? So we have to figure out in the passage where they talk about the RNA transport mechanisms. They mention RNA a bunch of times. So expression in RNA, delivery of RNA. That's kind of what we're talking about here, delivering RNA. So delivery of RNA to target organs has previously been challenging due to RNA's rapid degradation, but cationic lipids complex with RNA have allowed for delivery and expression of the complexed RNA. So we're looking for a cationic lipid to complex the RNA. Remember that lipids are these compounds that generally have a long alkyl chain, a really nonpolar area, and then something polar on the other end. So triglycerides have the ester groups, fatty acids, when deprotonated, have the carboxylate group. You can have ammonium groups, and then we have compound C. This is actually a carbohydrate. So we'll roll that out. That's not a lipid. Then it's really just a matter of finding the cationic lipid. This one's neutral. This one's anionic. D is cationic. And that should be our correct answer for that one. The next question asks us by what mechanism is a nucleic acid carrying liposome most likely to be taken into a eukaryotic cell during CNAD. CNAD is their abbreviation for cationic lipid-mediated nucleic acid delivery. So we look at these different processes. Well, exocytosis is transport out of the cell. Symport and antiport refer to transport that involves specific channels that transport small molecular entities. Endocytosis involves transporting things that are encapsulated with these membrane shells around them and into the cell. So that would be the only reasonable answer in this context. Our next question is which approach would be the best component of gene therapy for a patient for whom the treated condition is expected to persist throughout the patient's life, even after therapy. So we look over at the passage and we find that there is a mention of chronic conditions. This is something they do in the MCAT a lot. Chronic conditions, and over here it says conditions expected to persist throughout the patient's life. Two things that are synonymous with each other but stated in a different way, so that you have to correlate them with one another and find them to match them with a passage. So once we've matched that up, we can look at that sentence, and it says CNAD is preferable to viral DNA delivery in those clinical applications that do not require long-term expression for chronic conditions. But we are being asked which one is necessary for chronic conditions because this is expected to persist throughout the patient's life. So CNAD is preferable to viral when it's not chronic. This is chronic, so viral delivery is probably the preferable method for persistent or chronic conditions. And next we're asked about a very specific cationic polymer. With which units in DNA would the cationic polymer, shown here, most strongly interact in a supramolecularly assembled packet of cationic polymer nucleic acid? And we don't actually need the passage other than it telling us that cationic polymers and lipids can coordinate to nucleic acids. We can see that this has a positive charge in the backbone, and if it's going to have an attraction of any kind with a nucleic acid, it should be attracted to the negative parts of the nucleic acid. Purine and pyrimidine bases, while polar, are neutral, but backbone phosphates have negative charges carried in one of the oxygens in each repeat unit, and the deoxyribose sugars are very polar but not ionic. So anionic groups will have the strongest attraction for cations through an ion-ion interaction. So moving on to passage 5, we see we have some enzyme kinetic data here. And the first question is, what type of inhibitor can we conclude the added compound is on the basis of the Lineweaver-Burke plot shown? Now there are a lot of different metrics you can kind of read off of a Lineweaver-Burke plot. One thing we need to do to answer this question correctly is reading the text. We can see that one plot for the enzyme and the enzyme with various concentrations of added inhibitor are shown below. So the control with these little circles, the blue circles, is the enzyme with no added inhibitor. As you add more and more and more inhibitor, increasing concentration, the rate slope changes. And we see that all the lines intercept on the x-axis. That is characteristic if you look at the enzyme topics in the MCAT Ladder course. That's really characteristic, diagnostic for non-competitive inhibition. They next ask you again to interpret this kinetic data plot, and they're asking you to find Vmax, the maximum velocity, the maximum reaction rate of the uninhibited enzyme in these units. It doesn't really matter what the units are, but you see the units that they're using are on this axis. The units here are in concentration of reciprocal concentration of substrate. The uninhibited enzyme, again, is the one with these little circles. And one thing you can tell from a Lineweaver-Burke plot, again, if you look at our enzyme kinetics topic, is that this point right here where you cross over the y-axis is equal to 1 over Vmax. So we have to solve for Vmax with 1 over Vmax is approximately 0.002. So we'll pull up our whiteboard, and we know that 1 over Vmax equals 0.002. So Vmax is the reciprocal of 0.002. So we'll bump our unit over 3 spaces. We have to bump our decimal unit over 3 spaces here as well. So our fraction becomes 1,000 divided by 2, which is 500. Going back to the question, happily 500 is one of the answer choices. You'll notice they've got 0.002 as a distractor, but you'll have to know that's a reciprocal of that particular unit. It even says 1 over V in this article, so that could even cue you in, but don't rely on that being there. Just you'll have to know that that point where it crosses is 1 over Vmax. All right, so the passage is about adding these inhibitors to the enzyme. And one of the things they have in here is this structure. So this question says, the authors of the study provide this generalized schematic for effective MAO inhibitors. MAO is the enzyme they're talking about if you read the passage, where AR is to be an aromatic group and E is ideally a resonance electron donor, so an electron-rich zone. Based on this generalized molecular composition, which would be expected to be the best MAO inhibitor by the mechanism under investigation in this study? So one thing we can do is we can look for the aromatics. The 6-membered rings with the alternating single-double bonds are aromatic, whether there's a nitrogen or not. But a cyclohexane is not aromatic. That would let us rule out compound C as being the correct answer. The second part they talk about is this being an electron donor. Fluorine is an electron withdrawing group by induction. Nitrogen has a lone pair, oxygen has a lone pair, oxygen has a lone pair. Those would all be reasonable, actually quite good electron donors. So we have B and D. We've ruled out C because of the aromaticity not being here. We rule out A because these are electron withdrawing, and it says we have to have electron donors right here. So which one of these two is right? They both have aromatics and electron donor groups. Well, the last thing that doesn't mention the text of the question, but is in the picture, is that there should be a 4-carbon chain between the two. 1, 2, 3, 4, 1, 2, 3, 4, 5. So B is the only one that meets all of the criteria that the authors lay out for being a good MAO inhibitor. The last question associated with this passage says, oxidative deamination is the reaction catalyzed by MAO enzymes. Which of the following shows the product of oxidative deamination of alanine? Now, in writing this question, I was kind of nice, because the passage does tell you that MAO catalyzes oxidative deamination of monoamines. So this question could well have said, which is the product of alanine upon action of MAO? All right, and you'd have to hunt in the passage to find that. But this is how I phrased it. And it requires you to know a couple things. First, you need to know what alanine is. Alanine is actually A. So if you didn't know the structure of alanine, you guessed A, you would have guessed the starting material alanine, not the product. And then breaking down what oxidative deamination is, the name tells you that the amine will be lost. It's deamination. So these two, A and C, that have the amine, I hope you'd rule those out anyway. And then oxidative, you've got to oxidize the amine when you remove it. So this is alanine. If we look at the site where the nitrogen is attached, in this potential product, you've got a hydrogen there instead. Adding hydrogens is reduction. Here we've put 2 bonds to oxygen. That's oxidation. And choice D then would be the correct answer for deamination, losing the amine, and oxidizing at the same time. So here we are at passage 6, covering questions 24 through 27. And you see that we have some information given to us with this question. A plot of UV-Vis data as well as some structures. Let's go into the questions. Which describes the organic reaction of NBD chlorine with H2S to form NBDSH? The structures and these names they give to them are given down here, so we really only have to look at the scheme to figure this out. And if we zoom in a little bit on this, you see that the only change in going from NBDCl to NBDSH is the chlorine is substituted for an SH group. So looking at the choices, it's not an elimination reaction. An elimination reaction will lead to the formation of a new double bond, which we're not doing here. It's one of the substitutions, and the question is whether the reagent that's being used to cause the substitution is a nucleophile or an electrophile. So reading the passage, what you see is that it's exposure to H2S that causes this reaction to take place. So down here it tells us the sensing mechanism for detection of H2S relies on reaction of NBD chlorine to form NBDSH. So it's detecting H2S by reaction. H2S has a nice lone pair on the sulfur. So you could look at the periodic table and see that sulfur is in the same column as oxygen. If we draw out H2S, you have these nice lone pairs. Remember that a nucleophile donates electrons to something, so that would be considered a good nucleophile. This is what allows us to call this a nucleophilic aromatic substitution rather than electrophilic aromatic substitution. The next question asks us if the following were used in place of NBD chlorine as part of the H2S sensing strategy, which would absorb with a peak maximum at the longest wavelength. And they're referring to this type of experiment where you're looking at the absorbance versus the wavelength. This is an ultraviolet visible spectrum, UV-vis spectrum. So longest wavelength. Remember that the longest wavelength corresponds to the lowest energy. And in UV-vis, the more conjugated unit has the lower energy absorption. It takes the least energy to promote if you have a long pi conjugated system. And that should correspond to the longest wavelength. Remember that a pi conjugated system is alternating double, single, whether around the ring or in a chain. So you see there are 6 bonds that are sort of engaged in a system like that. But compound C has only 3 bonds engaged in that system. So A would be said to have a longer pi conjugated system, longer wavelength. But B has the longest pi conjugated system. It goes all the way around the whole outside of the molecule, and we lack the double bonds here. So looking for that longest consecutive strain of alternating single-double, compound B by far has the most pi conjugated system. So next we are given more data from this study, and it says the image below shows a plot of color change on the y-axis of a sensor film versus the concentration of added H2S. Which of the following is an accurate statement of sensor performance based on the data available? So the color changes from gray at the bottom to red at the top. So you can think of this as a measure of how red or purple this material has become. So here are the potential answer choices. Upon exposure of a sensor film not previously exposed to H2S, the film will not change color if it is exposed to a concentration of greater than 1.5 ppm. All right, if there is no H2S, the color is gray. If we expose the film to a concentration greater than 1.5, which is here, this answer choice suggests the color won't change, but clearly it's going to be changing to red. Right? These dots indicate that you've changed from gray to red. So that's not right. This says the sensor cannot detect concentrations of H2S below 1.5 ppm at all. So here's 1.5. It's red. Below 1.5, it can go from gray to somewhat red, eventually you reach the maximum amount of redness that you can get, or purpleness that you can get. So certainly we can detect below 1.5. In fact, it's very predictable how much the color will change. It's a linear relationship. So choice B is also not correct. There are only enough sensor molecules to react with about 1.5 ppm of H2S in this film. Well, let's see. If we have some molecules and we have 1.5 ppm of H2S, and then we keep adding more molecules, the color won't keep changing. That suggests we've saturated our system. That could be the right answer. But let's look at choice D. There is a linear response of the film at H2S both above and below 1.5 ppm, allowing quantification of H2S concentration across the full range given the x-axis, the y-axis value. So part of this answer choice is correct. There is a linear relationship here. There is a linear relationship here, but they're saying if we know the y-axis value, we can tell what the concentration is. But see all these dots here? The Y value is not really changing, but the concentration is changing a lot, from 2 to 3 to 4 to 5. So that choice D is also not reflecting what we're observing in the data. So choice C is the only one that really makes sense here. So then we have the last question associated with this passage, and it says, one of the mechanisms by which H2S is thought to exert toxicity in humans is by inhibiting cytochrome C enzyme activity. If this is the case, which process would be the most directly impacted in a patient following their exposure to H2S? So we kind of need to remember what the biological role of cytochrome C enzyme is. Hopefully you'll remember from our biochemistry study that cytochrome C enzyme is involved in the electron transport chain, which is part of the process related to oxidative phosphorylation. Cytochrome C enzyme is not one of the many enzymes involved in DNA replication or repair or the Krebs cycle. So the oxidative phosphorylation would be the best answer in this case. So here we are on passage 7. This will cover questions 28 to 32. And you see it's a relatively short passage, not laying out a whole study, but it's laying out a scenario where most of the information is really this passage. And it's an air gap capacitor made of 2 triangular plates, and you know their dimensions, so you can figure out their areas. You know how far apart they are. And they're telling you that you have a 50-volt battery, and it's connected to the plates for 30 minutes. The permittivity of free space, E sub naught, is 8.85 times 10 to the negative 12th farad per meter. So we have all the information we need to assess different things about this circuit. So the first question is, which best approximates the capacitance of the system? So you'll need to know the equation relating capacitance to the other metrics of the system. So capacitance is related to the area, the distance of the plates, and the permittivity of free space by this equation, we just sort of scroll out on the whiteboard here. Now we need to fill in the different parameters. If we look back at those parameters to figure out the area of the capacitor and the distance, and they give us the E naught, we can fill in those values. So looking back here, we see that they give us these dimensions in centimeters. 15 centimeters, 40 centimeters, 25 centimeters. But we need to use meters so that we can use the units of the permittivity of free space, and the SI units are needed for volts and farads and all of that. So we're going to convert these to meters. So 0.25 meters, 0.4 meters, 0.15 meters. One of the things we need to figure out is the area. So we're going to need to figure out the area of a triangle, which is half base time height. So with those things in mind, looking at this diagram and pulling up our whiteboard, we'd be kind of taking notes, writing these down as we were thinking about them. So you see I filled in the base and the height of the triangle, the half base height equation. The distance between the two is 0.25, and then we have the permittivity of free space that I've jotted down here as well. So talking it through, 15 times 4 is 60. But we have 2 decimal places over there and 1 there, so 1, 2, 3. So we're going to have 0.06. but times a half. So we're going to have 0.03 for our area. And we've already converted this to meters. The distance I've also converted to meters, 0.25 when we were talking it through. Then we have this epsilon, 8.85 times 10 to the minus 12th. I'm actually going to estimate that to be 9 times 10 to the minus 12th because I don't want to try to multiply out decimals while I'm trying to scurry for time in my exam. All right, so I've got these numbers plugged in here. I need to do this math. So I have 0.25 on the bottom. That's a quarter. So dividing by a quarter is the same as multiplying by 4. I'm going to go ahead and do that. You don't have to do this, but for me it's a little bit easier. So now I have 4 times 9 is 36. Times 10 to the minus 12th times 0.03. So I have to do 0.03 times 36. So 36.03. 3 times 6 is 18. 3 times 3 is 9. 10. So 108. But I've got to bump 2 decimal places over. I'm going to get 1.08 times 10 to the minus 12th that I still have hanging around here. So Looking at this 1 point something times 10 to the minus 12th, the units are in farads because we had everything else in meters to cancel out. And they give us the units like this. So 3 point something or 1 point something. We're very close to one of these two units. Nano indicates times 10 to the minus 9th, but we had times 10 to the minus 12th in our answer. Pico is times 10 to the minus 12th, so C should be closest to what we calculated. And there you can go through the math again. The second question associated with this circuit is if the capacitance of the circuit is 1.1 times 10 to the minus 12th farad, then the charge stored in the system is which of the following? So we need to relate capacitance C to charge Q. So the relationship we need is that charge is capacitance times voltage. We're told the capacitance in the problem, and we're told that the voltage of the source hooked up is 50 volts. So we need to do 50 times 1.1 times 10 to the minus 12th. Well, 50 times 1.1 is 55. So it's just 1 times 50 plus another tenth of 50, which is the 5, times 10 to the minus 12th. And if you're using the SI units for voltage and farads, it comes out correctly to the coulombs, the units of charge, and that's actually one of the answer choices, 55 times 10 to the minus 12th. So we got that relatively quickly, assuming we know that the charge Q equals capacitance times voltage. Question 30, given that the charge stored in the circuit is 55 times 10 to the minus 12th coulomb, then the electrical power stored in the system is which of the following? And power is energy per unit time. This is where the time factor, 30 minutes, is going to come in to play. The knowledge of capacitance, power, and energy we need is that power is energy over time, as we said, and energy is also equal to QV over 2. Now we know what Q is. That's the charge. We know the voltage. We have that in the problem. Time we also have is 30 minutes. Now you have to be really careful. Whenever you use time in science, you almost always will need to convert this to seconds. Right? 30 minutes times 60 seconds per minute will give us 1,800 seconds. So we're going to plug that in for T. The nice thing about having the whiteboard is that you can plug in these units and just cross out things or wipe them out. Next we have to plug in Q and V. Well, Q was 55 times 10 to the minus 12th. V was 50. Got these in the SI units already, so I'm not writing in the units. Divided by 2. Now we have to do this math to figure out the energy first, and then divide by 1800. So 55 times 50, I'm just going to estimate that's about 50 times 50. 50 times 50 is 2500. Divide by 2. All right, I'm going to pretend that this is 2600 because I changed my 55 to 50. 1300. Okay. I'm going to say this is 1300 times 10 to the minus 12. That's the energy. All right, so I can cross that out and put it there in that place. I have to divide that by 1800 still. So I can cross out the two zeros. 13 divided by 18 times 10 to the minus 12th. Look at these potential answer choices. I see that 0.76 times 10 to the minus 12th is pretty close to what I've got. 13/18 is around 3/4, which is about this answer. So I would have to guess that this would be the correct answer. So this question is saying, if a dielectric with kappa equals 25 is inserted in the gap between the plates while everything else is still sort of connected how they mentioned, which statement is true? Well, just quickly, if we look back at the equation we already needed to solve an earlier part of this problem, this is the case for this air gap capacitor. But if you put a dielectric in between, you have to multiply by kappa. Here, our dielectric is equal to 25. So we'll actually be multiplying the capacitance by a factor of 25. So if we know the equations just very quickly, we can figure out what type of answer we're looking for. So going back to the potential answer choices, capacitance is decreased, no. Capacitance is increased by a factor of 5, No. Capacitance is increased by a factor of 25. Yes. Capacitance is increased by a factor of 25. So we're down to answer C or D. The second part of the answers then is what will help us differentiate this. Now the capacitance is actually inversely related to the electric field between the two plates. We learned this in our MCAT physics course. So one of these suggests the electric field increases, one suggests the electric field decreases, so we just figured out that the capacitance does increase, so the electric field in that region has to be reduced proportionally. So choice D would be the correct answer in this case. Our last question about this particular circuit is if a dielectric is inserted between the plates of the capacitor, which quantity is unchanged from the original circuit? And of course the answer is going to be charge. We store the same amount of charge across the two plates no matter what we put in between them. The amount of time it may take to discharge or something like this may be influenced by the dielectric, of course, but charge is conserved. You cannot have a different charge built up across the plates just because you stick something different between the plates. So charge should be the answer, and that wraps up passage 7. Alright, so our next question is about this eye, which is going to be treated as an optical instrument. This is something that's laid out in the MCAT outline as a specific thing you ought to be able to do. And they tell us in this little brief passage various things about the eye. The first question about this optical instrument, number 33, is the tree in the image is 25 centimeters from the eye. Where is the image formed? So we need the lens equation to solve this. This is one of the important equations in optics. Optics is, I think, a high yield area to study getting ready for the MCAT in terms of physics. And this problem tells us the focus is 1.75. That's the 1 over f in the lens equation. That equals 1 over the distance of the object, which is 25, plus 1 over di, that's the distance of the image, which is what we're trying to figure out. So we need to solve for di. So 1 over di, isolate on one side, equals 1 over 1.75 minus 1 over 25. We need to be able to solve this without a calculator, so I'm going to actually change 1.75 using a trick. 1.75 is 1 3/4. That's 7 quarters. The reciprocal of 7 quarters, since it's on the bottom, is 4/7. I also know that 1/25 is 4% of something. So I'm going to put 0.04 instead of that. So you can look at the math tips and tricks to do this quickly without a calculator that we have on the physics and math review course. 4/7 is also relatively easy because I can just divide 7 into 4. And I can say, well, 7 goes into 42 exactly 6 times. This is about 0.6. So now I have a good estimate of 0.6 minus 0.04 for 1 over di. So 1 over di is about 0.60 minus 4 is 56. Now this is 1 over di. Got to be careful. So di is the reciprocal of that, 1 over 56, 0.56. That's about 1 over a half, which is about 2. So I've maintained all of my units in centimeters, so it should be 2 centimeters. So let's look at what the potential answers are. And fortunately we see an answer quite close to 2 centimeters. So we could have rounded in different ways, but this is the correct answer. The next question says that print in books is typically 3.5 millimeters high. What is the height of the text on the retina when the book is held 30 centimeters away from the eye? And here we need to use our knowledge of magnification, how the heights of the images relate to the distance of the image and object away. So here we have negative di over do equals hi over ho, where o refers to the object and I refers to the image. So if we take a look at this, we can plug in the known values for distance of image. So from the front of the eye to the rear of the eye, you have 2 centimeters. So we'll have the negative sign is in the equation, so negative 2. The object is 30 centimeters away. So So again, I'm not writing centimeters everywhere. I'm just making sure that I have centimeters as my units for everything. Then I'm looking for the height of the image. That's what I want to figure out. And the height of the object itself was 3.5 millimeters. So 3.5 millimeters, I need to convert that to centimeters. So 3.5 millimeters, there are 10 millimeters in 1 centimeter. So I'm going to cross that out. I'm going to get 0.35, leaving all these things in centimeters. All right, now the answers are given in meters, so we'll have to convert this to meters at some point. Just on your whiteboard, remember what units you're using so that you can keep consistent. Or you're going to mess something up by orders of magnitude and have it multiplied by some value of 10. So now I need to solve for HI. So I'll just rearrange 0.35 times negative 2 is 0.7 with a negative sign equals 30 HI. So divide by 30. So negative 0.7 over 30. I'm going to make this 7 into a 6 and just say that's about the same. So I have 0.6 divided by 30. If I divide by 10, 0.06. Now I divide by 3, negative 0.02 centimeters. This allows me to go and take a look at the answers and see how close I would get. Now I remember that the answers are in meters, which is 1/100th of this. So it's going to be negative 0.0002. 2 extra zeros to be 1/100th. So a meter is 100 times bigger than a centimeter. It's going to be something like this, 0.0002. The negative sign just tells me the image will be upside down, and the question doesn't ask me about that. The question just asks what its height will be, not which direction that height was pointing. So an answer of 0.0002 is what we got. That seems to match this answer choice. And you'll see they've written the answers out here in a variety of different ways, some of which are proper scientific notation, some are not, just as a way, a wrinkle, to evaluate your ability to move around decimal places. And choice C looks to be correct, and this is the equation we use to get there. Next we have a strand of hair, 0.1 millimeter thick, placed under a microscope with an objective magnification of 10 times magnification, eyepiece magnification of 5 times. How thick is the hair as seen at the eyepiece? Now this part of the problem does not ask anything about what the image is doing once it enters the eye. It's at the eyepiece. We don't need any of the information about the eye itself that might be looking in that microscope. So we have 2 pieces of important information. We're doing a 10-fold magnification at the objective and a 5-time magnification at the eyepiece. So overall we're doing 10 times and 5 times 10 times 5 is 50. So we're doing a 50-fold magnification of a 0.1 millimeter hair. So we would get 5 millimeters, right? Multiply by 10 first would get you to 1 millimeter, and then multiply by 5, and that's how I get the 5 millimeter relatively quickly using, again, some of the math tips we talk about in our physics course. 5 millimeters is answer B, and that is the correct answer for that problem. The next question isn't really asking us to do math, it's asking us to evaluate whether we know properties of certain types of optical systems. So which statement is false about the eye? One thing we need to know going in is it tells us the eye can be treated as a thin convex lens, creating an image of the object as shown here. And the answer to this one, if we read through the choices, it looks like it's going to be B, because we have a converging lens and it can be used to correct a defect where images of distant objects are formed in front of the retina. That's what this says. You're not going to correct a defect of distant objects formed in front of the retina with an eye. These other things are true about these general types of optical systems if you want to read through, just as sort of a memorization aid. But certainly you can't correct defects in that manner. So passage 9 is about the discovery of some different radioactive elements. And one of the questions is about uranium. Uranium is an example of a blank. Representative, non-representative, transition metal, or noble gas. Non-representative elements are the name given to elements that are not in the S or P block. So this is the transition elements in the middle, as well as the F block at the bottom of the periodic table. You could look at the periodic table to see that uranium lies at the F block at the bottom of the periodic table in the separated out strip. And that's the correct answer here. Next it asks to which of the following elements is radium most similar in terms of reactivity and properties. We'll pull up a periodic table to look at that. So we're looking for radium. We can find radium right here. It's asking which element it's most like, and we see that it's in the same column as barium, strontium, calcium, magnesium, and beryllium. Probably one of those elements. Going back to our screen, we see that strontium is one of the choices, and that would be correct. Next question asks about uranium most commonly found in the Earth's crust as uranium-238. That's how many neutrons are in this isotope. So we're talking here about uranium-238. 238 represents the mass. So that's the protons plus neutrons equals 238 is what it comes down to. And we see that uranium down here is number 92, which indicates there are 92 protons. So 238 is the sum of neutrons plus protons. If we do 238 minus 92, we see that we would get 146. As it turns out, that's one of the potential answer choices, and that's correct in this case. And the last question for this passage, which of the following elements contains a valence shell electron configuration ns2, np4? So S and P electrons adding up to 6. So pulling up our periodic table again, if there are 2 S filled and 4 P filled, that's enough to get us 1, 2, 3, 4, 5, 6 spots over and be in this column. Oxygen, sulfur, selenium, tellurium, polonium. Polonium is one of the answer choices and would be correct in this case. Now we're on to our very last passage of this section, the Chemical and Physical Foundation section. There will always be 44 questions associated with passages in the biological and chemical and physical sciences sections. This passage is about the blood buffer system, which is a really high yield type concept to think about and learn about for the MCAT. The first question is, if the pH of the blood decreases too far, becomes too acidic, an increase in breathing removes CO2 from the blood through the lungs, which way does the equilibrium shift? All right, so here's the equilibrium they're referring to. If you add acid, which way does it shift? Well, you have to recognize that this is the acidic species, and if we add something to one side by Le Chatelier's principle, it pushes the equilibrium the other way. So if we have it here, it should push to the left, and that will be the correct answer in this case. The next question is saying if the concentration of carbonic acid in blood is 0.0012 molar, how many grams of H2CO3 are present in 100 milliliters of blood? So we have to convert moles per liter to grams per 100 milliliters. So I've written out molar as mole per liter, first of all. I'm going to convert this to grams, so I need units of grams on top and units of mole on the bottom. So how many grams per mole will I get out of H2CO3? And I'm going to do this without a calculator, so I don't want to use all the decimal values. So each hydrogen is going to weigh 1. I'm just going to use the nearest integer. Each carbon weighs 12, and each of the 3 oxygens weighs 16. I get 48. Plus 12 is 60, plus 2 is 62. So I'm going to use 62 for my molar mass. And that'll be how many grams are in a whole liter of blood. It asks for 100 milliliters, but I'll work that out later. I don't really want to multiply 12 times 62. I'm going to pretend this is 60. So 60 times 0.0012. Well, I'm going to multiply by the 10 first. So I'll have 0.012 times 6. 6 times 12 is 72. So 0.072 moles in a whole, or the moles is gone, it's grams now, so grams in a whole liter. Now I need to convert this to 100 milliliters, so 1 liter is 1,000 milliliters. So how many grams of this are in 1 milliliter? If the grams will still be there, we will have 1,000th of this many. So I'll have to move the decimal place over 3 more spots. 1, 2, 3 zeros before the 0.72 that was already there. 0.72 grams in 1 milliliter. But they're asking for 100 milliliters, so I'm just going to have to hop back 2 spots to multiply by 100. So my answer should be 0.0072 grams. Go back to the answer choices. I see that I have one answer very, very close to what I had calculated. And turns out that's the correct answer, and you could go through the math as well. Next question is about the delta G of the reaction at equilibrium. So consider the first portion of the blood buffer system. What is the sign of delta G of the reaction at equilibrium? And this is kind of a trick question. Delta G at the equilibrium is by definition 0. This is just knowing that definition, that's how you define equilibrium in terms of the energetics. The last question is, what is the molecular geometry around the central atom in carbon dioxide? It gives a variety of different choices. Well, to look at the Lewis dot structure, we'll pull up our whiteboard. We'll have to draw the carbon with 2 oxygens. Try to give everything an octet. So the carbon has an octet through the bonds. Here are the lone pairs. Remember that you want to make sure you don't use too many electrons. So carbon, if you look at the periodic table, is in the 4th tall column. Oxygen is in the 6th tall column, and there are 2 of those. So we have 4 electrons from the carbon valence shell, 12 electrons from the oxygen valence shell, a total of 16. And how many did we use? 2, 4, 6, 8, 10, 12, 14, 16. So what kind of geometry corresponds to this Lewis dot structure? Well, if I have 2 pi bonds around a carbon, only 2 sigma bonds. 2 sigma bonds means that I have an sp hybridized carbon. 2 sigma bonds, s and p are the only 2 orbitals I need to make sigma bonds. I don't need to have 2p or 3p here. An sp hybridized carbon has a linear geometry. So we go with linear, and that's the correct answer here. That was the final question associated with a passage, and again, this little sidebar will come up. You can close that so you can just see the quiz. You won't need to refer to passages to answer the rest of these questions. Which fat-soluble vitamin must be produced by photochemical reaction in the skin? This is just sort of a memorization of what you've learned about the different vitamins. Vitamin D is the one that you need to have sunlight to produce in your own body. How many stereoisomers are there for a linear form aldohexose like galactose or glucose? Well, the rule we need to know is that the number of stereoisomers we can possibly get is equal to 2 to the n power, where n is the number of chiral centers. So if we have an aldohexose, as we have here, we need to figure out how many chiral centers there are, and then we'll have our answer. Remember that no matter what the aldohexose is, you have 6 carbons. And the aldo tells you there's an aldehyde at one end, and of course there's going to be a CH2OH at the other end. That uses 2 of the 6 carbons, so the other 4 along the backbone can at most have 4 centers that are chiral. All right, so 2 to the 4th power is 2 times 2 times 2 times 2 is 16. Go back and plug that in, and that turns out to be the correct answer in this case. Next, which best describes the functional groups present in the product of reacting S,4,6-dihydroxy,5-ketohexanoic acid with an excess of pyridinium chlorochromate? This is a good example of where you might not need to spend as much time as you think drawing the structure of this complicated molecule. We have hexanoic acid. So let's start by drawing that on our whiteboard. Draw a quick 6-carbon chain. The carboxylic acid has to be at the end of the chain. Go back to our problem. At the 5 position, there's a ketone. At the 4 and 6 position, there are hydroxys. So 4 and 6, hydroxy, 5 ketone. 1, 2, 3, 4, 5, 6. 6 position and 4 position have hydroxyl groups. 5 position is a ketone. All right, so there is a chiral center here, but the question is asking us about oxidizing this with PCC. If we're going to be using PCC to do a reaction, we don't really care about R or S, so we're not going to waste time drawing a specific stereoisomer if the answers are just what functional groups are present. So PCC will take only one hydrogen away from a carbonyl. The carbonyl is the name for the carbon that has the OH on it, so take that one H away. Every H that's taken away has to have a new bond to an O to make up for that lost hydrogen. So we'll lose this hydrogen, and the oxygen will of course not have too many hydrogens, so it will be neutral. So it really is as simple as that when you have PCC to do this reaction. And what we see in terms of functional groups are we have an aldehyde here, we have 2 ketones here and here. One was already there, but nothing happens to it. And then we have the carboxylic acid. Let's look back at our answer choices and see if aldehyde, 2 ketones, carboxylic acid is one of our choices. 2 ketones, 1 aldehyde, 1 carboxylic acid happens to be answer choice B. It is right in this case. Next we have what is the definition, or which is true rather, of an exergonic reaction. So we need to know the definition of exergonic. The exergonic, endergonic type terms, the gonic refers to the Gibbs free energy, thus the G, to help us remember that. Exer means releasing heat. So exergonic would mean the favorable heat releasing spontaneous reactions. So delta G should be negative. The reaction is favored in the forward direction when you have that case. So whether something's exergonic or not doesn't tell you how fast it happens. It just tells you the energy involved in thermodynamics of stability of products and reactants. Concentration of reactants equals the concentration of products because the reaction is at equilibrium. No. The concentration of products exceeds the concentration of reactants. For a spontaneous exergonic reaction, that will certainly be the case. All right, so number 49, which is an example of a structural difference that allows differentiation of RNA and DNA. And if you read through these answer choices, the backbone saccharide in DNA has one fewer oxygen than RNA is the one that makes sense, especially knowing that D stands for deoxyribonucleic acid, RNA ribonucleic acid. So there's one fewer oxygen in the saccharide ring. Number 50, which term best defines coenzyme? This is just a case of remembering what the correct definition is. The coenzyme is necessary to activate the enzyme, but it's not itself an enzyme, so it's not the term for the enzyme that needs a cofactor. It's not the term for an enzyme that needs a cofactor and has the cofactor bound. It's usually a small molecule that is needed to bind. All right, so that's the definition for coenzyme. Now we have a physics standalone question. A toddler pulls a wooden horse of mass 3 kilograms on a cement floor with a force of 5 newtons, at which point the horse just begins to move. The coefficient of static friction is 0.5. The force of static friction is closest to which of the following? Now, we might be tempted to break out some equations here, but remember that the force of static friction is defined as the force you need to apply to just get the item to move. Once it starts moving, it's not static friction anymore, it's kinetic friction. So the 5 newtons is given to you in the problem, and it's just a matter of knowing the definition of the force of static friction. So here we are in 52. What is the buoyant force exerted by water on an object of volume 2,000 cubic centimeters? And when we look at these answers, only 2 of the answer choices have units of force. Kilograms is units of mass, so right away we can tell it's B or C. And we have 2,000 cubic centimeters. So let's break out our whiteboard. We have 2,000 cubic centimeters. And one thing we know about buoyancy force is that it's proportional to the volume of water displaced. And the volume of the object completely submerged is the same as the volume of water displaced. But we need to know the mass of that water displaced. Well, water has a density of 1 gram per cubic centimeter. So if we have 2,000 cubic centimeters of water, that's 2,000 grams of water. Now we need to get from mass, which is what we have here, to force. How do we do that? Well, force equals ma. And generally we get force in units of newtons as the answer choices are given. And the a, the acceleration, remember, is that gravitational acceleration. All right, so I need to get this in units of kilograms, not grams. So there are 1,000 grams in 1 kilogram. Divided by 1,000, I have 2 kilograms of water displaced. 2 kilograms times the gravitational acceleration is 9.81 meters per second squared, is about 10 times 2. So our answer should be about 20. Looking back at our problem, we see that 20 is one of the answers, and that would be the correct answer in this case. Number 53, when steam rises up from the surface of a hot spring, what kind of heat transfer mechanism to the surrounding air occurs? Well, the heat transfer is from steam, which is a fluid. Remember that liquids and gases are both defined as fluids. When you have heat transfer involving a fluid, convection is the general mechanism by which heat transfer occurs. Question 54. A jet flies at a speed of 1,000 meters per second, while the speed of sound in air is 345 meters per second. Which statements are true? Well, you've exceeded the speed of sound. And these answer choices have to do with hearing the sonic boom. You'll only hear the sonic boom, of course, if the jet exceeds the speed of sound. The sonic wave is always heard after the jet goes by because of the cone that trails behind the jet, which needs to reach the ground, occurring after it's passed the observer. So you have an answer choice, the observer on the ground hears the sonic boom after the jet passes them, would be the correct answer in this case. Number 55, what is the maximum number of electrons that can fill in the n equals 5 shell? Let's get our whiteboard out. Luckily, there's a really simple rule for figuring out how many electrons a shell can hold. A shell can hold 2n squared. 2 times n squared, where n is the shell. Here the shell number is 5. 5 squared is 25. 2 times that is 50. So go back and check our potential answers. We see that's one of our options, and that turns out to be correct. Number 56, we consider the Lewis dot diagram below. If atom X were to become an ion, what charge of ion would be the most stable? Remember that atoms tend to pick up electrons to become a closed shell or octet-bearing species. This has 7 electrons. It would pick up 1 more to get to 8. Adding 1 extra electron to a currently neutral would make it -1. And that would be the correct answer here. The next question is, which would be the closest approximation of the pH of a 1.2 millimolar hydrochloric acid solution? So we'll have to know the definition of pH, and it has to do with the concentration of H+ ions. We'll have to know that hydrochloric acid is a really strong acid that completely dissociates, and we'll have to know how to convert units of concentration. So here's our definition of pH. pH is negative log of the concentration of H+. H+ here is equal to the concentration 1.2 millimolars, because that's the concentration of hydrochloric acid, and hydrochloric acid will completely dissociate to form H+. 1 H+ per HCl. So So we're going to have to convert our 1.2 millimolar to molar. So that's the units we have to use in here. 1.2 millimolar, milli indicates 10 to the minus third, is going to be 1.2 times 10 to the minus third molar. So our pH is going to be the negative logarithm of 1.2 times 10 to the minus third molar. Now this 1.2 is not very big, it's pretty close to 1. So to make my logarithm easy to do, I'm going to do negative logarithm of 10 to the minus third molar. Now the logarithm of 10 to the minus third is minus 3. Remember, the log is just the power you need to raise 10 to. So minus 3, but negative log plus 3. So we should be kind of close to 3. We see that 3 is in fact one of our answers, and that would be correct in this case. Number 58 shows us a chemical reaction, asks us which type of reaction it is. This is going to be a case where you have the metal with chlorines on it, and the metal with sulfates on it, and both metals switch with both anions. That's an example of a double displacement because you've displaced both of the units. And finally we get to our very last question of the chemical and physical foundation section, which is the correct expression of equilibrium for the reaction below. And it's always products over reactants. So we have to have products on top, NO2. Looks like they did that in all these cases. The coefficient becomes a superscript. So it should be NO2 squared on the top. And then you should have N2O4, the reactant, on the bottom with no superscript. Well, it kind of looks like A and C both have the right units. So what's the difference between these two answers? We have a Kc or a Kp. C indicates concentration, P indicates the use of partial pressures. These are both gases. When you deal with gases, you generally deal with the partial pressures, not the concentration. So we're going to choose choice C, and that will round out our CPF section. Now, once you have gone through this on the MCAT Ladder program, if I advance this, it will tell me what my score is. And I've gone back and made some of the answers incorrect so that I can show you the symbol for an incorrect answer. Right? We got all the correct ones as we went through, of course. But it'll tell you what your score was, so you know where you're standing on the chemical, physical, foundation type problems. And it'll give you this red X if you got the question wrong. It shows you the question and the explanation typed out. And here the right questions are going to be the green check marks. So you can go back and review all of them. It's always a good idea. There may be extra information in the explanation that helps you learn additional information. You can also, of course, have the passages. It'll let you scroll through all the passages. So to look at passage 1, so I got question 1 and 2 wrong on passage 1, let's say. Then you can look at the passage and look at the explanations and try to figure out where you want to stray with those. And I guess I didn't miss too many of these, because I went back and just made a few of them wrong. Remember, we actually got 4 V1 when we did this one, but I went back and changed the answer, so I got this wrong. What would I have had to do? And it shows us the math. Once you get down to the end where you don't have passages anymore, you can close up the passages if you'd like and just look at the questions and review these standalone questions as well.